Hello. How are you guys doing? Hopefully my face should pop up in a second and some other bits will happen and magic is happening. Hey. I'm always slightly surprised when it actually works, that everything ends up being exactly where I'd expect it to be. How are you guys doing? I need to turn off my sound on my laptop. Hey, Darren. Darren, is there a timetable for these? Yes. So essentially what's going to be going down is I'm going to be doing one of these sessions every Wednesday. So Wednesday, 5 o'clock, I'm going to be doing a biology session. I believe that chemistry is tomorrow. And I don't know if maths and physics are doing one, but I'm sure the person who is writing stuff and comments to me will be able to let you know if anything is going down. So how many people are watching? 12 of you watching. Guys, feel free to say hi if you're on the chat. Just got your bio mock results and got a B. Oh, Aisha, well done. That's really good. Well, unless you're not happy with that, in which case it's not good, but hopefully you are happy. Hey, Goldchin. Hey, Shik. How are we doing? How are your days today? Anyone do anything remotely fun? I guess some of you probably have only just got back from school or college as well, which is sad. Hey, Megan. Hey, Darren Chipmunk. Is this on photosynthesis? Yes, it is on photosynthesis, like the big thing in front of me says. Cool. You're watching a movie with your friends. Very nice. You're in year 13 as just. Okay, cool. So today I'm just basically planning on going over photosynthesis. So people tend to get quite stuck with this. And from our Instagram poll, which some of you may have taken part in, this was the thing that I think most people were upset about. So the kidneys was really big, photosynthesis was big, and respiration too. So I reckon that we should get cracking. I'm going to go over the light dependent and the light independent stage, and we'll do some exam questions too, and see how we're doing. Yeah, Goldchin, don't worry, everyone finds this hard, I think. I think you just need to go through it enough that you understand it. So essentially, no worries. Essentially what we're going to look at then is we're going to be talking about where photosynthesis actually occurs. So we're going to be talking about the specific places, because that's a really standard exam question of just where it happens. We're going to look at the light-dependent reactions and the light-independent reactions. And there's nothing really that I'm going to miss out here. So in case you are interested, and I'm probably not going to leave this up for very long, but feel free to come back to this at some point, because this will be on YouTube forevermore, to have a look at what you're expected to know. And I would literally just go through it being like, yes, I know that. Yes, I know that. Yes, I know that. No, I don't know that. I'll spend some more time going over it. There's OCR for those of you that do OCR. Very, very similar. The only difference, in fact, no, I don't think OCR does have any differences, actually. Apart from AQA don't care about non-cyclic and cyclic, and OCR do. The only big difference here, which I'm going to address later on, is that TP is called GALP if you do EdXL. I don't really know why, because they're talking mostly about something called triose phosphate, or I guess they're calling it glyceraldehyde phosphate. But just so you know, if you see GALP at any point or TP at any point, they both mean the same thing. Okay, I'm going to try and keep mentioning that over and over again anyway. So starting with a light-dependent reaction then, I think it's firstly really important to realize with this that you have more than one pigment. Does anyone know why we have more than one pigment? And Layla, you're in the right place. Hey, T-boy. So why do we have more than one pigment? So in our thylakoid membrane, in fact, I should probably talk about this. I'll talk about it in a second, guys. And you guys tell me, why do we have more than one pigment out of interest? Different wavelengths, what do you mean? I'm not letting you get away with simple answers. To absorb different wavelengths of light. Yeah, lovely. So as you guys probably know, there is lots of white light which is constantly hitting plants. And you guys will also know that white light contains lots of different colors, but it collectively looks white, right? So here are some different wavelengths of light, or some visible light anyway, which will be constantly hitting plants. Plants tend to have more than one pigment because if they didn't, they would only be absorbing certain colors. So chlorophyll A and chlorophyll B are only absorbing wavelengths of light in this range here, and also over here too. Right? So there needs to be something called xanthophyll, which is another pigment, and carotene, which is another pigment, just to essentially ensure that as much of the light that's hitting these plants as possible can actually be used. Because it would be a complete waste of time if you could only absorb like a certain color of wavelength if you can't then use other ones, because I don't know, you're just wasting that amount of light that's hitting you. So what are we saying? No clue, Darren. Hopefully no. Hi, Ollie. Yep. You guys are all good. So just so we know then, we have multiple pigments to absorb as many wavelengths as possible. If you scroll up on the comments as well, there are some slides that you can have so you can fill this in at some point. But to absorb as many wavelengths as possible. Now, the two places, or the two really important places that it's really important that you are aware of are the thylakoids and the stroma. And you might see that more than one thylakoid, if they're stacked on top of each other, are called grana. Well, that's the plural. Singular would be granum. So all of this sort of stack here, which I've unintentionally left off here, this is a grana. Ooh. My laptop is not very good at writing at the very bottom of the screen, the bottom left. But anyway, in terms of where different things happen, do you guys know what goes on in the stroma and what goes in the thylakoids, out of interest? I'm glad that you understand this this part of it anyway. Plants in general, people find quite hard, I think, because we're very far removed from plants. Will I be doing epigenetics? I can do epigenetics if that's something that lots of people want. Stroma is light dependent. No, other way around. There you go. So the stroma is the light independent and the light dependent is on the grana. Good. So in the stroma, which is essentially cytoplasm, and just because I've seen an exam question saying what is different between the cytoplasm and the stroma, with the answer being the enzymes there are slightly different and the molecules there are slightly different, like lots of TP and GP or GALP or whatever. But the stroma is where the light independent reaction occurs. Whereas the thylakoids, or the grana, or whatever you want to say, this is where the light dependent reactions happen. There you go, light dep. Why is there non-cyclic and cyclic photophosphorylation? British girl, interesting name. I will get to that in a bit. Okay, so there are two types and it depends on essentially where light is hitting, but I will talk about that, so don't you worry. So if we're looking at non-cyclic then, I've always tried to teach this so that you have it as simple as reasonably possible, right? So with non-cyclic photophosphorylation, all you will ever need is a diagram that looks like this. If you can draw this very quickly in an exam, you'll be absolutely fine to remember this. So I've always taught my students just to remember this, or I have just drawn this out so many times I've remembered it when I was in your shoes. Okay, so if we're going to talk about this, I think it probably makes sense to start off talking about how light is involved. So does anyone know the first thing that's going to happen? So what's the first thing that's going to happen in the light dependent stage? Why is photosystem 2 first? I don't actually know. It just is. It tends to be one that we put first. So photolysis, okay. Pigment absorbs light energy, okay. Photolysis of water, okay. Right, so I wouldn't start off with photolysis, which I think is controversial, because if we start off with it, the whole point of photolysis is to replace electrons. So I would actually start off with some light coming in and hitting photosystem 2. Now, depending on your spec, you either need to know that photosystem 2 is this thing that contains chlorophyll, or instead of saying PS2, you can just say chlorophyll. It doesn't actually matter too much. It depends on your specs. It matters a lot. You don't know what I'm saying. So the very first thing then is a photon of light is going to hit chlorophyll. So if you want to know that that's in PS2, fair enough. And by PS2, I mean photosystem 2, in case you haven't heard of that. So a photon of light is going to hit chlorophyll. Okay, so you either need to know that or you don't. What happens after a photon of light has hit chlorophyll? Can anyone tell me? I'm just going to stare at you until you come up with a good answer. Very quite annoyingly, I have to look over here because that's where my comments are coming up. Light hits, yep, photosystem 2. Good. So this will excite some electrons. Okay, so electrons are excited. Now, there are a few other words that go along with this. And depending on your spec, you'll need to know this. And I'll tell you what they are. So you can either talk about chlorophyll over here as being photoionized. So you could use that if that's something that your spec wants you to know about. To be fair, it is still scientifically true, I guess, even if it's not in your spec. But chlorophyll is photoionized. That is one thing that goes on. Or if it's going to lose an electron, what do we call that process? So what's the process called when we're going to be moving when electrons are lost or gained? Does anyone know? Good. So you could say that chlorophyll has been photoionized, or you could say that it has been oxidized because it has lost an electron. Okay, what happens to that electron? So here is my electron, which has been lost. It's actually two of them. It's oxidation, sorry. So don't forget oil rig, yeah? So oxidation is lost, reduction is gain. So chlorophyll has lost an electron, so it's been oxidized. What happens next? Where does this electron go? Someone said it. It goes through electron carriers, or it goes through the electron transport chain. Right. To save having like hundreds of little proteins, which I could have drawn, I'm just too lazy to. I've just drawn one. So this little electron is going to be basically moving through this electron transport chain. And again, depending on your spec, you'll either need to know that it goes to PS2 or it just goes down the electron transport chain. Okay, so I'm going to say it moves through the electron transport chain to photosystem 2. Right, some of you don't need to know that literally that last little sentence. Does this mean it's been oxidized? So yeah, essentially what's going to happen then is as it moves through, it's going to go from, it'll join with one protein, which means that that protein's been reduced, which means that the one before has been oxidized. Then it will go to another one. So that protein's going to be oxidized and the next one will be reduced. And you basically have this continuous like redox reaction happening where one protein is reduced and another one is oxidized as these electrons move. So I know lots of you are now telling me that this is really important because it means that hydrogen is going to start to move. And that is totally true. I'm just going to ignore that for right now. I should probably say as well that this is going to be my stroma. And this is going to be, if I get rid of oil rig, oh, that was sad. This is my inner membrane space, or whatever your spec calls it. They're all the same. So essentially, I would need to have, I will eventually have protons moving through, but I'm going to ignore that for now. The next up that I want to talk about, and I'll explain what non-cyclic means as well in a second. Don't worry, I've got another very similar slide after this one. Essentially, what's going to happen now is water needs to be split. So your chlorophyll, your P680 chlorophyll, does not like that it has been oxidized. It is very, very unhappy about this whole thing occurring. So light can also hit a molecule of water, which is called photolysis. Okay, and when light hits water, it will turn it into some protons, it will turn it into some electrons, and it will turn it into half an oxygen. Okay, and then you can balance this differently if you want. And I also need to have 2 of each because there's two H on the left, so I need 2 on the right. If I've got 2 positive things, I need 2 negative things. There you go, that balances. You can turn it into 4, 4, and 1, but it really doesn't matter. But anyway, these little electrons, which I've just generated here, these are going to go back. Ooh, my bad. Oh, whew, I lost it all. These are going to go back, not through the electron transport chain, just happens to be in the way, but they're going to go back to P680. They're going to go back to my photosystem 2. So we can say for number 4, photolysis of water is going to occur. So photolysis of water is going to occur. And apparently I didn't actually mean to write this here. So photolysis of water is going to occur. This will replace electrons. Okay, so the electrons which have been lost from PS2 are now replaced by the splitting of water, essentially. Okay, right. Anyone got anything that they are confused about? What loses the electron? So chlorophyll loses the electron. It does pump hydrogen. I'll get there in a second. What do you mean by non-cyclic? So non-cyclic means it's not going in a circle because these electrons are going to PS1 and it's not going straight back to PS2. But it's only PS1 actually involved in cyclic, but I'll get to that in a second. In stage 3, doesn't electron go to PS1? Yeah, so when it moves through the electron transport chain, it goes to, very good point, I've written the wrong thing. It goes to, apart from getting rid of it, there you go, it goes to PS1. So my electrons have basically just moved on. Now, if you ever get asked a question about why photolysis happens, people get confused with this. All you need to say is it replaces electrons to chlorophyll. That is the whole point of it. So if we carry on from here, then we know that we've got these molecules or these electrons moving about. Another thing that also happens is light is going to hit PS1 as well. So light is going to hit PS1 and some electrons are going to be excited to a photon of light. Okay, so some electrons are going to be made here because it's been hit by light, and these electrons are going to get used a little bit later, and you guys might remember why that's the case. Oxygen being the last acceptor, that's respiration, nothing to do with photosynthesis. So following this then, I said earlier that you've got these electrons which have been excited. Oh, I need to stop leaning on my keyboard. You've got these electrons which have been excited. And some of you guys rightly told me that these electrons, as they move down the electron transport chain, they're going to sort of give off some of their energy. So not all of their energy is going to be used in this process. They're going to lose some. And this energy is used to pump hydrogen ions. So there will naturally be hydrogen ions in the stroma, which is down here. And these hydrogen ions are going to get pumped through not the electron transport chain, but through certain carriers within the membrane which can transport hydrogen using the energy from the electrons. Okay, so electrons are going to move through the electron transport chain, and this is going to provide some energy to allow hydrogen to move through. So we can say that hydrogens move through. To the membrane space, or the inner membrane space, as you may have heard it. Okay, so what's going to happen next then? So I've now got a whole load of hydrogen ions up here. What's going to happen next? So where are they moving from? They are moving from the stroma, which is this guy over here, and they're moving to the inner membrane space. So what happens next? Lovely. So the next thing that happens then is we are going to form this massive concentration gradient, which essentially allows for hydrogen to then start to diffuse through. And there are a few words that you might have heard for this, such as chemiosmosis or electrochemical gradients or something like that. So we can say that a high concentration or a high conc grad or electrochemical, and by grad I mean gradient. I'm going to write gradient. I feel like I've been lazy now. So electrochemical gradient of hydrogen ions or protons is established. They move through ATP synthase. Okay, when they move through ATP synthase, essentially what will happen is this whole molecule will basically spin. It's really cool if you ever go on YouTube and Google what this thing actually looks like. But as hydrogen moves through it, it basically starts to spin and it causes ATP to be produced. So you can say there's like a conformational change or something along those lines, but you end up making ATP by reacting ADP plus inorganic phosphate makes ATP. Okay, so this is sometimes called the chemiosmotic theory. So electrons have been excited, they've moved through the electron transport chain, And this has used energy to pump hydrogen through into the inner membrane space. They've established this big concentration gradient. They've moved through this ATP synthase channel, and essentially ATP has been formed. Okay, what else could we say about this? Yeah, let's say that this is called the chemiosmotic theory. In case you've ever wondered why it's an electrical chemical gradient as well, as opposed to just a chemical gradient, it's because you have like a chemical gradient of a high concentration of hydrogen up here and a low concentration down here. But it's electro because you've got a big positive charge at the top. And as you guys know, like charges attract, opposite charges repel, other way around. Opposite charges attract, like charges repel. If you've got a massive amount of positive charge at the top, they're going to naturally repel each other. So you've got sort of two actions helping these protons to move. Where does the energy come from to pump the hydrogen out? It comes from just electrons moving down the electron transport chain. So they're literally just moving down and some of their energy is lost, which allows for hydrogen to be pumped through. So moving electrons, you can almost think of it as being electricity. It's almost like there's electricity moving through this membrane, which is then used to pump hydrogen. So it's literally like you've turned a plug on. What else are we saying? Someone's late. Don't worry about being late. You can watch it from the beginning as well. Is it a hydrogen pump? It is a hydrogen pump, yes. So what's the final thing that we need to talk about then? So I've had electrons moving up here, moving through the chain, coming down here, coming to PS1. Some of these ones have been excited and they're going to be used a bit later on. Hydrogen has moved through. It established concentration gradient. What's the final thing we need to talk about? Does water get broken down in the stroma? No, we tend to talk about photolysis happening in the inner membrane space. Good. Formation of NADP and ATP. So we've already made ATP by hydrogen moving through ATP synthase. The last thing that happens is all of these electrons need to get used in something. So essentially what we have is a molecule called NADP. I've always remembered that the P stands for photosynthesis. It definitely doesn't, but it might help you remember this. This is going to react with the electrons plus the protons to form reduced NADP or NADPH. Okay, so the very last thing that we need to be aware of is all electrons and protons react with NADP to reduce it. Yeah, so reduction is always the gain of electrons or the addition of hydrogen, pretty much. So it's doing both. It's gaining hydrogen ions and electrons. And if you have hydrogen ions and electrons together, that is basically just hydrogen. So NADP is reacting with hydrogen to make NADPH. Catalyzed by the enzyme NADP reductase. Yeah, you are totally right. So that is all you need to know about non-cyclic photophosphorylation. That is literally everything. So all you've got to do, and I know I'm saying all you've got to do is just remember that it starts off with some lights hitting the chlorophyll, that generates some electrons, they start to move through the chain, that provides energy. You get this like concentration gradient, it moves through. And I think the last thing to remember is just what's happening to all of these electrons and protons. Like they're not just going to sit around, so they have to react with NADP. Right, so I would recommend from now on that to remember this, you just draw this little diagram out and just draw everything that happens. Like it's really not very hard. Like you can draw that in about 10 seconds, probably less than that. So good question, T-Boy. What is the difference between cyclic and non-cyclic photophosphorylation? Essentially, when we have cyclic photophosphorylation, it only involves photosystem 1. And your light energy, your photon of light will hit chlorophyll. It will excite your 2 electrons. They will move down the electron transport chain. And instead of going anywhere else, they just come back to the same chlorophyll molecule. So it's constantly going round in one cycle, right? This is really, really useful in the sense that it still generates some ATP. So if we go back to this, if this electron is, if we've got some light hitting PS1, my electron is going to be moving up. It's going to move through the electron transport chain, so hydrogen can still get pumped through, but then it goes back to chlorophyll again. If I've got hydrogen going through, it can still produce ATP. So PS1 is used. It produces a little bit of ATP. But the main difference that you need to remember is that no NADPH is formed, right? The reason for that is because the electrons are all going back to chlorophyll. So if they're going back to chlorophyll, they can't get picked up by NADP, right? So no NADPH is formed. And to be honest, that is one of the more important elements of this. I think arguably it's the most important part of this, because you need to eventually donate hydrogen ions. You need to be able to reduce things later on. Okay. So no reduced NADP is made, but ATP is made. And that's the difference you need to remember. This is always seen as like a really minor little thing that doesn't really have much detail put into it. But just remember that ATP is formed, no NADPH is formed. Okay, right. How are you guys feeling with that? I know people find this confusing. Can they happen at the same time? Yeah, they will be happening at the same time, but non-cyclic is better. How are we feeling, guys? If you feel perfectly happy, send me a 1 or a smiley face. If you find this okay, send me a 2. If you find this really confusing, send me a miserable face or something that displays your, I don't know, hatred of photosynthesis. Darren Chipmunk, if you want to tell me what you're confused about, and Malika and Toy World of Honey, let me know what you're confused about. What's the point of having both? Because one of them makes ATP. Oh, they both make ATP, so this is going to lead to as much ATP as possible being formed. Okay, I think more of you are confused with this as possible, which means I'm not actually going to move on. Guys, all you need to remember, look, this is exactly how I would do this in an exam, right? I would have a little drawing of what I want. I'm stubborn as well, by the way. So just remember, you have electrons being made. So photoionization first. Electrons are made. They move through the chain. Lovely, right? You guys know that these electrons are ultimately going to get used later on to join with NADP, right? You know that for anything to actually happen, you need protons to move. So the protons are going to use the energy of the electrons to form a concentration gradient, right? If you have a concentration gradient, you guys will know that that's going to diffuse through ATP synthase, which leads to ATP being made. Final thing to remember, NADP plus hydrogen ions plus electrons equals NADPH. Right, that is pretty much all there is to this. Don't get too caught up about cyclic photophosphorylation. Non-cyclic is the thing that people like to question the most. Oh yeah, sorry, and photolysis of water. How did I forget? So photolysis of water is also the thing that replaces these electrons. So any chlorophyll molecule doesn't like having its electrons taken away, so it needs them taken back. So photolysis is going to happen, which is going to make some electrons, it's going to make some protons, and it's going to make some oxygen as well, but not a lot. How does hydrogen ions create ATP and ADP and pi? So, or inorganic phosphate. So the hydrogen ions are just moving through this enzyme called ATP synthase. And when a proton moves through it, it just generates ATP. So a chemical reaction will happen, which causes ADP to be reacted with a phosphate group, which produces ATP. In the absence of light, less electrons are excited, so it's more efficient to have non-cyclic, as ATP can be made with less light. Yeah, fair enough, if there is less light. Yeah, honestly, that's all you need to do. Just draw this little diagram out in an exam, and you've got all your answers that you need to really think about. I know I'm biased in saying that this works, but I promise you this is how I learned it and have remembered it after all this time. Right, so let's see how we're doing then. So describe the part played by chlorophyll in photosynthesis. What does chlorophyll do? I'm gonna wait for you guys to tell me some answers. It's the kinetic energy of hydrogen going through ATP synthase that generates ATP. Yes, exactly. Absorbs light energy. Good. Mark 1 is just for saying absorbs light energy. Good start. Next. So some of you are talking about this. Okay, I need more of you to tell me the right answer before I say anything else. Good. So it excites a pair of electrons. So in fact, I'm going to reword this because I've done that poorly. Let's say light excites electrons. Where do these electrons go? Provide a green pigment, and that is why plants are green. The pigment just reflects the color green. So they pass into the electron transport chain. So this is correct. That's a mark. That's a mark. If you talk about the electron transport chain, that doesn't actually get you a mark, but I'm going to keep it there. So electrons move through ETC, electron transport chain. Don't be lazy in an exam like I'm being. What else do we need to say? This is year 13. What else do we need to say? So photolysis of water will occur. That is true. Annoyingly, it's also not a mark, but you're right. And why not just say everything you can? Photolysis of water replaces electrons. The last mark of this is actually really stupid, if I'm totally honest, because it's not really too much to do with it. So yeah, hydrogen ions pumped. That's still not a mark. Chemiosmosis happens. Go on, what's the point of chemiosmosis? And you'll have your last mark. Good, lovely. Final mark, in caps. There you go. I think it's a bit of a bold statement to say, describe the part played by chlorophyll in photosynthesis and that they produce ATP, but on this mark scheme, Maslow's got the mark. So if you see a question like this, you're going to have to be a bit general, I think. Just say pretty much everything you can. Please explain the topic immunity. Abdul, I'm not going to explain the topic immunity when I'm talking about chloroplast and photosynthesis. Maybe another time. Allows ATP to be produced. Lovely. Right. Describe the membrane. Sorry. Describe how the membranes inside the chloroplast are involved in photosynthesis. How would you answer this? So describe how the membranes inside the chloroplasts are involved in photosynthesis. Abdul, I'm not going to say the same thing 3 times. They contain the reaction center. Good. I like that. Tell me more. I'll tell you when you get marks. Good. I'll definitely give a mark for that. So contain ATP synthase. That'll definitely get you 1 mark. What else could we say? They have a large surface area to do this. Yeah, I like that. I like that you said that. It's not actually a mark, but I think it's a good point. They have photosystems. Yep, equally, that's the same mark as this kind of. In fact, yeah, let's say that. So contain photosystems / chlorophyll, depending on your spec. What else can we say? Primary reaction center, yes, yes, yes. Absorb different wavelengths, yes. Thylakoid membrane is where the light dependent stage occurs. Lovely. Right, yeah, that is the final mark. Well, not the final mark, but that's another thing you could say to get your 3 marks. So the site of light dependent reaction, you could say that. Nova, you are totally right to say that. No one ever says that, so well done. It compartmentalizes where some enzymes and some molecules are. So it means that the molecules that need to be within the inner membrane space can't just move out. So it compartmentalizes too. You could get them up for that. You could say that the space in between the membrane provides a site for hydrogen ions to build their concentration. You could say photophosphorylation, that'd get you a mark. I think that's about it. Non-permeable to hydrogen ions. Yes, I guess that is a good point, but I guess you want to say, no, actually, no, yeah, I like that. I think I'd give you that, Aisha. Light harvesting system, yeah, all of that, that's like talking about the photosystems. Yes, I would give that to you. Right, moving on then. So we've done the probably harder bit. This is the easier bit in my mind. So the light independent reaction then. So we are specifically talking about what is going on in the stroma. Okay, and the products of the light dependent reaction are used to synthesize hexose sugars. So by hexose sugars, I mean sugars which have 6 carbons, such as glucose, such as sucrose or something like that. Okay, yes, so you guys are totally right that this has a name. So what are you saying? The Calvin cycle, Krebs cycle, which one is it, Calvin or Krebs? Lol, not Krebs. It is Calvin. So yeah, this is often called the Calvin cycle because the guy who discovered it, I guess discovered it's the right word, the guy who kind of worked out how this works, and it was a guy. And as is always the case in any science that happened like over 50 years ago, it was an old white man, which isn't the case anymore, but certainly was back then that they seemed to do all the science, which is probably a problem with society at the time. Anyway, the Calvin cycle is what the light independent reaction is. And we're going to say that this uses NADPH and ATP. That's all I'm going to say here. And then this is the Calvin cycle, right? This is what you guys need to know about. So how could we describe it? What's the first thing that happens? Tips of preparation in 2 months. Abdul, I'm talking about something. I will give you some tips at the end if you want. How is GP formed? Go on. RUBP joins with CO2 to form and go. Okay, I don't quite understand. CO2 combines with RUBP, catalyzed by the enzyme Rubisco. Good. Yeah, really, really good. Is it the same person who owns a Calvin brand? Unfortunately not. No, I can't remember his name. It's something, I swear he's got like a litter. What's the word? What's the word when you got like the same letters? Alliteration. I think he's got an alliterative name. It's like Calvin or something, which isn't quite right. But anyway, Malika, I'll get back to you in a second. So first thing we need to say then, CO2 is fixed to RUBP, ribulose bisphosphate, by the enzyme Rubisco. The most abundant enzyme in the world, that one as well. Good little fact. Is it John Calvin? That's really, is it Melvin Calvin? Oh, I remember it being something funny. Anyway, so when you do this, CO2 fixes to ribulose bisphosphate by Rubisco, and you actually make this really weird 6-carbon molecule, but it's only very, very brief. So it's very unstable. So very quickly, it will break down to form 2 molecules of GP. Okay, so we can say 2 times GP produced. Right, I'm going to keep this one really basic this side, and the next side is going to get a little bit more complicated. What happens to those 2 GP molecules then? It is Melvin Calvin. Oh, I knew it was a great name. Thank you, Snap Revise, for telling me that. Good. How does it form TP? So what are the 2 reactions that happen? So TP, in fact, let's turn this as well into GALP. For those of you doing Edexcel who are screaming at me at the moment, this is called GALP. There you go. TP/GALP is produced by what processes? What's it called when it when ATP breaks down? And what is reduced NAD given? NADP as well, to be specific. You can see I've caught you out. You guys are trying to tell me that it's ATP being hydrolyzed. That's not what I'm talking about. What are the 2 names when a phosphate group is donated and when hydrogen is donated? Hydrolysis is definitely not what I want. If I add hydrogen to something, what's it called? Good. Phosphorylation is one word, produced by phosphorylation. So ATP is going to provide a phosphate group. Phosphorylation and reduction. Good. Well done, guys. I'm impressed. So TP is made by reducing and phosphorylating GP. Hydrogenation, isn't it decarboxylated? No, it certainly isn't. Decarboxylation is when you lose a carbon. Look, there's 3 carbons there, 3 carbons there. Nothing has been decarboxylated. That's probably good that your teacher has taught you. It's not that hard, to be honest. People make a bigger fuss out of it just because there's lots of steps, but it's not too bad. Okay, final thing then, what happens? TP is going to get converted into what in this cycle? I know there are obviously this step, but I'm going to talk about this in a second. What happens to TP then? Finally, what goes on with TP? Turns into RUBP. How is it turned into RUBP? What is the name? Look at what's happening. Don't tell me about this step here. I know you're all right in saying hexose sugar is made, but for now, bear with me. How is ribulose bisphosphate made? What is happening? What happens to it? What is added to it or whatever? So RUBP made by, what's it called? Guys, have you all forgotten the word that I told you a second ago? Phosphorylation, that's the word I was looking for. So if ATP has been broken down into ADP, then TP has been phosphorylated. So RUBP is made by phosphorylation. Okay, this is technically called substrate level phosphorylation too, for those of you that have done respiration. So all you need to remember for this, and I'll get to the hard bit in a second, is if ATP is broken down, then whatever that molecule was there, that's being phosphorylated. So it's having a phosphate added to it, right? If hydrogen is added to it, so the same thing is going to happen, but hydrogen, then it is being reduced. Reduction is the gain. Oxidation is the loss, reduction is the gain. If you gain hydrogen, you're being reduced. Okay, so that's sort of the more basic bit done. If we have another go at explaining the stuff that's going down, or if we have a go looking at the numbers as well, can you tell me, and these numbers here are really arbitrary, right? They're sort of just different points to make more than anything else. So what happens to TP then? What's the important thing that happens to TP? I'm going to talk about numbers in a second. Aisha, you are right. Finn, you're also right. What happens to TP? Someone tell me, before we start going all into the numbers. TP is converted into glucose, good. So TP is the thing which is turned into glucose. It's also used to make amino acids, ultimately. It's also used to make any other hexose sugar. Do you know if GP is used for anything, any of you? I've actually got the wrong way around, actually, haven't I? Sorry, my bad. I'm pretty sure that this one, the TP is used to make lipids as well. And the other one, GP, I'm going to give it away, can be used to make amino acids. My bad. Right. Okay, so let's look at these numbers then. Snickers get some nuts. Nice. So can anyone tell me, why are you all telling me that it needs to have 6 cycles? Because I know lots of people who can tell me that off by heart. And I know lots of people who can tell me that 5 out of 6 of these TPs are needed to make one molecule or to make my RUBP. But I don't know many people who can explain why this is the case. So why does this need 6 cycles out of interest? Probably a little bit before 6, but yeah, roughly. So RUBP uses 5 from GALP and 1 remains. So yes, true. 6 carbons for glucose, true. So, okay, let's have a look at this one. So for every carbon dioxide that I have going in, for every 1 carbon dioxide going in, I'm going to make my 2 molecules of GP and ultimately my 2 molecules of TP. Right? So if I want to make a hexose sugar, I need to have 6 carbons. However, the problem with that is I can't just take all of those sugars out, because if I do, I won't make any RUBP and this will just stop. So I can't just make my 1 TP, or I can't just make my 2 molecules of TP, sorry, and turn that straight into hexose. So why is it the case that I need 5 out of 6 molecules of TP to make my RUBP? So why is that the case? So let's say at least 2 TPs needed for 1 hexose sugar. But why is it that 5 out of 6 are going in to make an RUBP? So it is a 5 carbon compound. That is a very good point. So 1, 2, 3, 4, 5. So why is 5 out of 6 TPs needed? Sorry. So you need 12 TPs to turn 6 times, yes. Do you want me to show you some magic? Look at this. So if I've got 1 molecule of TP, I've only got 3 carbons. That's not enough. If I have 2 molecules of TP, then I've got 6 carbons. And again, that's not a 5 carbon molecule. So again, that's not really good enough. If I do another turn, 1, 2, 3, 4, 5, 6, I've got 3, 6, 9, 12. 12 carbons, again, that's not quite enough, right? Because if I've got 12, that's 5, 10, and 2 left over. If I do it again, 1, 2, 3, 4, 5, 6, so 3, 6, 9, 12, 15, 18. If I've got 18, again, not really ideal. Is my maths right here? Oh, I might be messing this up. So essentially what we need to realize is in order for us to get the number of carbons to be perfect, we need to know what number both 3 and 5 can go into. So what's the first number that I can make that both 3 and 5 can go into to get this right? Yeah, 15. So I need to ultimately make 15. I need ultimately to have 15 carbons here. So how many TPs do I need to end up getting 15? So if I want to get my 15, how many TP molecules do I need? Good. So I need 5 molecules of TP to make enough to have enough carbon left over to make RUBP. So 5 TPs will have enough carbon for ribulose bisphosphate. And then I can have my one other additional one, my 6th one, to make to leave me with 1 molecule of TP left over. Now the problem with this is if I've got 1 molecule of TP left over, hexose sugars are made from 2 TPs, right? So in order to make any hexose sugar at all, I actually need for all of this to go around 6 times, so that I'm left over with 12 TP molecules as opposed to 10, and that way I can make plenty of my ribulose bisphosphate and have enough TPs left over so I can then make my hexose sugar. Okay, so all in all, I need 6 turns to make 1 hexose sugar. Okay, so in case you've never wondered why this is the case, then you essentially need to have 6 turns so that I've got my 5 different sets of TP being made, or my 5 like 3 molecule things, so that I can then make 3 ribulose bisphosphates and just have a little bit left over. So that's why it's the case that you need these 6 turns and you end up with 5 out of 6 being used to make your RUBP. So all of this means that if I've got 2 ATPs over here and 1 over here, then I need 6 times 3, which is 18 ATPs. And then I've got 2 NADPHs over here. So 2 times 6 is 12. Just to make 1 hexose sugar. My maths was confusing. Yeah, I think my maths was a bit confusing there, sorry. Essentially what I'm trying to say is if you want to have enough carbons, so if I want to make something which has 5 carbon molecules, this cycle needs to work in a way so that you don't have any leftover carbons. And ultimately the way to do that is you're going to need to have 5 molecules of TP, so 5 times 3, which will give you 15. And 15 divided by 3, because RUBP is a 5 carbon compound, 15 over 3, over 5, sorry, is going to give you 3 molecules of it. So for every 5 molecules of TP, you can make 3 molecules of RUBP, if that makes sense. So 5 times 3 is 15. 15 divided by 5, the number of carbons here, is 3. And then you have to have 1 more cycle to end up getting your final TP molecule left over. Okay, how do you guys feel with that? I know that is a bit confusing. So 6 turns of the cycle to form 2 molecules of TP, which you can make 1 molecule of glucose with. Could you not just say that? I could have just said that. Yeah, Jackie, that is a very good point. But I wanted to explain why it's 5 out of 6. And I wanted you to think about how all these carbon molecules sort of come together. Aisha 1.9. Damn. Finn, tell me what you're confused about in a second when I show you this exam question. 1.999 is pretty close to 2. Yeah, agreed. Right, guys, have a look at this question then. And those of you that are confused, feel free to just write a message in and I'll try and answer it in a second. So the diagram below shows some of the steps involved in the process of photosynthesis. So place a cross in the box next to the name of molecule P in this diagram. So molecule P being over here. What molecule are we talking about? I'm waiting for a few more people to say some stuff. Yeah, good. So the products of a light-dependent reaction are ATP and molecule P, which is reduced NADP. Lovely. So molecule R and molecule S, what do we think they are? So comment out here. So what are the things needed to make ATP? So what do I need to make ATP? Lovely, well done. So ADP and inorganic phosphate, or you can call it a phosphate ion or a phosphate group or something like that. That is fine. Well done. So the photosynthetic cells contain many chloroplasts. Complete the table below by naming the part of a chloroplast where each of the reactions R, S, and T takes place. So R ADP plus inorganic phosphate makes ATP. Where does that process take place? It happens at ATP synthase, yes. What part of the cell, what part of the chloroplast is that happening in specifically? Good. So that is the thylakoid membrane. Well done. What about RUBP plus CO2 producing 2 GPs? So where is GP made? Lovely. And where is GALP made, or TP, for those of you who are AQA and OCR who are screaming at me saying, what's GALP? Lovely. So yeah, that is also a straight line. Well done. Okay, so suggest how GALP formed by reaction T can be used to synthesize the cellulose in plant cell walls. So starting with, well, not really starting with this, I'm sort of giving you a red herring now. What can we do with my GALP to turn it into cellulose? What's going to be going down? Do OCR need to know this? Yes, of course. Just change the word GALP to TP. There you go. So how is TP or GALP used to make cellulose? So it does regenerate RUBP, but that's not the bit I want to know about. This question is actually a lot easier than you think it is as well. So, Han, where is glucose coming from, British person? When did carbon ever get removed in that diagram? I don't see any carbon get removed anywhere. Nothing to do with carbon being removed. Something to do with beta glucose, yes. How do I turn, or where does glucose come into this though? Definitely do. We definitely should have some idea. Glycosidic bonds are involved. You're going to get marks eventually, but I do need to say, good. T-boy, you've told me. So TP is converted, or GALP, whatever one you want to talk about, is converted to glucose. Out of interest, how many molecules, so what percentage of TPs are used? So out of all the TPs that we have, how many of them are used to make glucose? Who has remembered the horrible maths? So 2 TPs are needed, yes. Thank you, Aisha, that was what I was looking for. So 1 out of 6 TPs used. Or you could say 2 out of 12. I guess I can get simplified, so it seems a bit stupid, right? Not 50%, because most of them are used to make RUBP, aren't they? So I've got this 3 carbon molecule, and I want to ultimately make something which has 15 carbon. It has 5 carbons. So 3 of them is not enough. If I put another few in, you know exactly what's going to go on. I'm going to keep doing this until we end up with the perfect amount, right? It happens to be 5 of these leading to 3 of these, right? If I have 5 TPs or 5 GALPs, that will lead to 3 ribulose bisphosphates with none left over, and then 1 more little turn can end up making my hexose sugar that I can then use. Anyway, so the rest of what you guys were saying is all right. So GALP is used to make glucose, and it's 1 in 6. You know that beta glucose is the one that is needed. So beta glucose is made. That'll get you a mark. Bam, bam, bam. Someone said earlier that we're going to need glycosidic bonds between them. Between the glucose, and then that is 4 marks if you want. But then final thing, if I've got, why beta glucose? Because beta glucose is used to make cellulose. If it was starch, it'd be alpha. Final thing we can say is hydrogen bonds form forming, making, then we could say something like, I don't know, straight chains, or it's not the straight chains, it's the thing that holds them together. So forming like microfibrils or something. Right, and then that's pretty much everything you can say. There you go, Aisha, exactly what I said. You could say this is a condensation reaction. That will get you a mark as well in this. You could also talk about 1,4 glycosidic bonds because they're long straight chains, as opposed to 1,6 where you get branches. Awesome. Right, guys, I am going to tell you something which I'm sure you are all dying to hear about just before you all head off. In case you are interested, for those of you who have seen my web classes, essentially what we at SnapRevise are doing in the next, I don't know, month or so, just before you have your exams, we have some seminars which are going on, which I am inviting you guys all to. So at Imperial University in London, there I am. No, I didn't have a beard then. Basically at Imperial University, we're going to be doing some seminars, and I am going to be covering a whole load of stuff. So in one of them, I'm going to be doing cell division, mass transport in plants and animals, Q&A sessions. You guys are going to be able to write messages in on your phone, and we'll have people who will literally pay and sit there to answer your questions. But essentially, I'm going to be doing 2. I'm going to be doing a year 12 one, and I'm going to be doing a year 13 one where I'm doing photosynthesis and respiration, inheritance, DNA technology, so the epistasis stuff and all the confusing bits, and some like complicated questions. But essentially, these are going to be in April at the very start, so sort of around Easter time. And yeah, the tickets are quickly selling out. So if any of you are remotely interested, feel free to go onto this website, which is snaprevise.co.uk/seminars. And I think there is a link coming up. And yes, I know it is £99, and I know it is expensive. I totally agree. But you are getting about 7 hours of education, which is pretty hardcore. There you go. I also believe that there is a discount thing if you want to go to maths, physics, chemistry, biology, where you can get quite a lot of money off, which I think it says something at the bottom of this page. If that is something you want, you can basically email us, and then we'll sort it out so you can get like a discounted rate essentially, so you can get them free. If you tell your school as well, I'm sure your school would be sort of looking for things like this to book up, because they like for basically days out at the end of the year, as I'm sure you guys all know. Where did I go to uni? I went to uni in Southampton for my degree, and then my graduate degree, I went to UCL. Anyway, guys, what you should now know before I run over any more than I should do, hopefully, is you should know the structure of the chloroplasts. So you should know about the stroma, and you should know about the thylakoids. You should know everything there is to know about the light-dependent reactions. So your electron movement, your proton gradients that you make, ATP synthesis, NADPH synthesis. And you should know about the light-independent reactions, where ultimately you're making TP, GP, and you're using 5 out of 6 to reform ribulose bisphosphate. Then your leftover ones are used to make glucose. Oh, right. Any questions, guys? Thanks, I forgot your name. My name is Ollie. No worries, though, Jackie. I'm glad that helped. What should you do if you need to learn the whole of AS and A2 biology in 1 year? Darren Chipmunk, I would get my head down and start revising very quickly and join SnapRevise and have more of these sessions. So Snap Provider, I'm doing like these sessions, we're in 4 of them a week or 5 of them a week at the moment. So if you wanted 5 hours of tuition, I don't think it costs very much, 5 hours. Anyway, I bought the premium package, but I don't know how to contact tutors, and I don't get any notifications for any web classes. I'm so confused. I don't really know how to use this. Malika, if you email, who can I get you to email? In fact, the person who writes in my comments for me, I'm pretty sure he would tell you exactly who to email. If you email, I don't want to write my email address down in case those you start emailing me. There'll be like a help section on the Snap Provide site. I'm sure you'll be able to write an email there and it will get through to someone who can then help you out. If not, you should be able to just write comments on the videos and it should come through. I don't know why that's not. But yeah, Malika, if you want to email Snap Provide, if you go on their website and find their help section, you'll be able to email them. Oh, Jackie, that is very nice. Do you have to know about ferredoxin? Depend on your spec, yes. How often do you make the live streams? I do, as I just said, 5 of them a week at the moment, which is what the ultimate package that we offer gives. Right, anyway, I reckon now is a good time to go because I've got another web class to do in about 40 minutes. So hopefully I'll catch you same time next week. I'm going to carry on doing the like tricky topics. So those of you that are on Instagram, if you follow Snap Provides, we do like polls every now and then, and I'm basically just working my way through the hard topics. There you go. I knew that was a email address somewhere. Right, gang, I'm going to head off. So have a really lovely evening, all of you, and I will catch you at some point in the future.